So good afternoon, class, and welcome to the fall semester. Thanks for enrolling in this course, Biotechnology and Society, BIO211. So before I go into the syllabus or the course, I would like to little bit share myself or my experience, my professional journey with you all. My name is, as you might be knowing by now, is Shiraz Mujtaba. I am a professor in the Department of Biology, of course, in the Medgar Rivers College. I am in this college since 2013 and 14. I did my PhD in the area of HIV, molecular biology and immunology. My work was mainly to study the immunological factors and various viral variants in AIDS and HIV positive individuals. After I finished my PhD, I got a postdoc position at Mount Sinai School of Medicine in New York, Upper East Side. And the reason I decided to do this postdoc at this place is because I was working with AIDS patients and HIV positive individuals, and I used to see various clinical spectrum in various patients. So it was very, very interesting, it used to raise my curiosity that the virus is same in every individual. Why does every other individual has a different kind of symptoms? And that's exactly what you see here in COVID also. Same virus, but it can cause nothing in some people, and it can be very, very serious in some other individuals. So that was, so since I was working with patients, I cannot go into detailed investigation because of ethical reason, but that was one of those interesting points that I wanted to continue working on my postdoc. And in my postdoc, I discovered the mechanism by which HIV replicates, how its replication escalates in AIDS patients. So after doing my postdoc, at a crossroad, what should I do? Should I go and join a company? Should I teach, or what should I do? That happens, once you graduate, once you finish one task, you're always, empty-minded. You're in a vacuum that what to do next. So, I decided to go for academics, and I had my own research lab until I was there at Mount Sinai till 2013. Then I came to Medgar Rivers College, the Department of Biology. And here I do teaching, administrative work, and research. And most of my research is published with students. So this is what has been my background, and this is what I'm doing right now. So with this brief introduction, and I just want to quickly add here that you have to introduce yourself on the discussion board, and your introduction mark you as a present. Remember, this is an asynchronous system of pedagogy. You don't have to come to the class, but you have to do the class assignment on time. On discussion board, your introduction is tagged with your activity in this class, and that is related to your enrollment. So make sure you do that as early as possible. So with that, let me go to the syllabus. So I would like to share with you my Blackboard page. So on my Blackboard page, I go to Biotechnology and Society. I click this, and here I am. So this is the page where I announce about class that will be meeting every Tuesday at between 2 to 4 p.m. Depends upon the how much long the meeting could be. This is my recurrent Zoom meeting across the semester. I'm not going to give you new link for every class. This is a recurring link, and I understand that by now you must be an expert with online education. Now, there are various folders here. All the teaching activities or learning activities will be posted in the content area. I will post all the quizzes, tests, and exams in the content area that you can see here once I announce the quiz or exam. Now, let's go to discussion board. So, I see almost every student has responded. So there are only, I believe in this class, there are 30 or 31 students. So I see that the total number of participants that have responded is 26 or 27. So 4 students have not responded. So, I cannot mark them positive or let's say present, not positive, rather present for this class. And, that could have, various kinds of consequences on enrollment. So make sure you introduce yourself in this discussion board by creating a thread. So with this, let's go to the content area. In the content area, you see this folder, which says BIO211 Syllabus Fall 2021. I will be posting all the learning activities so each week we will accomplish a chapter, and each chapter will be posted in this as a content folder. And you click on the content folder, and you see here is the syllabus. And before I go to the syllabus, that once I'm done with today's class, I will convert this recording into a YouTube segment, and then you can watch this video also in case you have not attended this class. Now, so let's click on the syllabus. So I'm just trying to save the Word file. I've uploaded the syllabus as Word file. I should upload as a PDF. So let me just go back to the Blackboard. So in the Blackboard, I go here and I say build content item, and I say that PDF copy of the syllabus for 2021. And I go and upload this syllabus. And I make this syllabus available to you until December 31st, although I think final exams would be between 15th to 20th December. So now let's go back and discuss the syllabus. So if you can see this syllabus. So here, we are meeting online, so you have the Zoom link. Right? And we are meeting on Tuesday between 2 and 4. I will be here at 2 p.m. And it's possible that all the students could come, but it's possible that none of the students will come because of asynchronous nature. So I will wait for the students until 2:30. And sorry, this class on Mondays, yes, Tuesdays are on between 2 to 4 p.m. I'll wait for 2:30. If students come, then we will interact. And if let's say if I'm not here, then you can always email me. My email is smushtaba@mec.cuny.edu. and I try to respond as quickly as possible, or within 24 to 48 hours. I respond to emails, to your students' email. So this is a course on biotechnology and society. This is designed to introduce students to the tools that biological or biomedical scientists they use. So these tools are used by scientists to develop products. Now these products could be a vaccine, this product could be a medicine, or these products could give rise to some kind of a conceptual knowledge. For example, if we don't understand the conceptual knowledge of DNA, we cannot do recombinant DNA technology. And if we don't do recombinant DNA technology, then we cannot produce a vaccine, right? So the message I'm communicating to you that fundamental knowledge of anything is important to make it applicable. If you understand something, theory, the basics, the fundamentals, then you can apply your knowledge. So that the nature of this course is that to give you fundamental knowledge of biology, and also also share with you what are those important tools that scientists have developed until now and what are their utilities, right? So this is a very interesting course which covers basic knowledge, applied knowledge, as well as some knowledge of regulation, right? For example, you must have heard last week or in a couple of days back that FDA has authorized Pfizer, has approved this vaccine, right? So why did FDA do? What does FDA stands for? FDA stands for Food and Drug Administration. What is its job? It is like a regulatory body that looks at the data, that looks at the quality control, it looks at the safety. So so any vaccine or any product that goes into public domain has to be verified in the hands of FDA. And if they give approval, then it goes to public. In case of COVID, because of the pandemic, it was given an early approval, but now I believe they have got the Pfizer. They have got the complete approval, which is a big thing. It is why it is a big thing, because it has never been before that in 1 year a vaccine has been made and has been given to millions of individuals in this country as well as abroad. So why it is that possible? That is possible because of technology. Where does the technology comes from? Technology comes from grassroots knowledge, the fundamental knowledge. So this is what is the nature of this course. Now I am very, it's very important that we discuss what are the student learning objectives to this course. Student learning objectives guides me to make sure what I teach you, and it also sort of gives you a path, a template that you will be expecting from me to teach you during this course. Especially, what I meant by course is biotechnology and society. Just a second, somebody wants to get in. Okay. Yeah. So I was saying that let's look at the student learning objectives or course objectives. Number one, discuss the definition of biotechnology. So definition is what I just mentioned to you, to expand fundamental knowledge, to develop a tool or a product, and this product can help human health and society, and also this planet, because the umbrella of biotechnology has enhanced. Rather than just doing with vaccines, it now affects climate change, right? Plants are very important. Some people say that plants are the first and foremost chemical engineers on this planet Earth. Chemical engineer or biochemical engineer. So biotechnology covers not only human diseases or human health, but also environment. There are a lot of pollutions going on is happening in this industrialized world. And where does this pollution go to? It is consumed by living organisms, including humans, including plants, including aquatic animals. So if we are dumping garbage in ocean or rivers, it is going to affect the organisms that are trying to survive. So biology affects environment, and the way to clean up is to employ new innovative technology. What is increasing in today's date is population. So it will continue to increase and the resources will shrink. So how to regenerate or how to generate new resources for growing population. So that's a broad definition and the scope of biotechnology. History-wise, the history of biotechnology goes very, very early, and there could be many examples. But one of the examples that comes to my mind, or what I read, is fermentation. So remember, in early years there were no refrigerator, but people used to consume milk or dairy product, and because there were no refrigerators or no tools to store food, these dairy product would convert into, let's say, what we consume as ferment into yogurt. And people used to consume yogurt, very back, a long time back. So that's a point of biotechnology. Similarly, fermentation of rice give rise to ethanol, alcohol, which is being used now as beverages in various, all over the place. So these are the earlier evidence that some of the earlier evidence that shows that biotechnology or fermentation was being done in very, very early stages of human civilization. And the fundamentals of modern biotechnology. So what are the fundamentals of modern biotechnology? So the major fundamental is to improve human health, improve the health of planet, improve the health of plants, and then when you improve these things, what is happening is that there are a lot of microorganisms. For example, what was not there in 2019, in 2020, we have to encounter viruses. What is the guarantee that there will not be any other new virus strain in next 6 months, or a variant of COVID, right? Now we are dealing with Delta strain, which is a variant of COVID. So, and then the COVID vaccines, what is the technology that they use? They used RNA-based technology, right? Which was never used before. So, these are the aspects of fundamentals of modern biotechnology, is to improve the planet. But there are enormous challenges that comes from our lifestyle, our population increases, many factors. So we will continue to discuss the definition of biotechnology in various aspects. History, as I mentioned to you just now, and the fundamentals of modern biotechnology. Point number 2, demonstrate the knowledge of the basic biology of prokaryotic and eukaryotic cells. So it is, as I mentioned to you, that we'll be talking about cells as a fundamental knowledge, but why we are talking bacteria? We are talking about bacteria, the prokaryotes. It is because most of the knowledge of biology we have gained comes from working on bacteria. Bacteria has been used as a lab to generate vaccines, to generate antibiotics, to generate various kinds of products. So it is very important to learn about bacteria and compare to human cells, how they differ. Because human cells are very difficult to culture, to maintain, unless there are cancerous cells. Okay, So we talk about this. Then we talk about, so if you look at our body is made up of cells. Cells are made up of chemicals, right? What do I mean by chemicals? The major macromolecules that supports a cell is protein, lipid, carbohydrate, nucleic acid. So what is DNA? DNA is a polymer of nucleotide. What is a nucleotide? It is a complex molecule of carbon, hydrogen, oxygen, and phosphorus, nitrogen. So, so chemistry is very important, right? So we talk about structure and function of DNA, how the information flows from DNA, how it goes to the various corners of the cell, how does cells function, right? So that's SLO number 2. SLO number 3 is discuss and identify the basic principles and common terms of recombinant DNA technology. As I mentioned to you, fundamental knowledge, if we have sound fundamental knowledge, we can develop some tool. If we don't have knowledge, then it will crash, right? Right from, when scientists or individuals, they saw a bird flying, they imagine of humans to fly, and they developed various kinds of gliders. Now you have planes, which are running at a very faster speed. So similarly, knowledge of DNA, then how does DNA relates to disease? How can we cure that disease? Can we manipulate the DNA? How can we manipulate the DNA? Then bacteria, how we can use bacteria to escalate, how to set up a biotech company to make a product that can be provided to the society. So we'll learn these things in the recombinant DNA technology, which is your SLO number 3. SLO number 4 says, discuss recent advances in biotechnology. For example, generation of genetically modified plant. What is the strategy there? Why do we want genetically modified plant? The answer is very clear, because the resources are shrinking, the prices are rising, the prices are continuing to rise. So how do we match up to this increasing expectations? And one of the ways is to take use of science, right? To although when the scientific development happens, there are controversies also from both the sides, which is very fair, because unless we debate, unless we discuss, we don't know the merits or disadvantages. So we look into all these aspects and try to understand what are the implications, and are the implications bad? If the implications are bad, do we still need it? For example, cell phone, right? It is there with us. An electronic gadget is with us all the time. We need it. We are dependent upon it for communication, 24/7, right? So, so you can debate, right? Is it good or bad? Similarly, scientific development, modern medicines, vaccines. Some people are taking vaccinations. Some people are debating taking vaccinations. So that's how, I don't know, I can't comment upon that, and I should not, but that's what happens. When technology takes a leap, there are views associated with that leap, and people can be on either side. And as a student, as a teacher, we are supposed to educate ourselves, right? And my intention is not to generate a debate, but my intention is to generate awareness, right? Demonstrate the knowledge of positive impacts of biotechnology on society. Yes, there are both kinds of impacts, but mostly positive, right? There are a lot of controversies about genetic modified plants, but so far I haven't read any article that says that, okay, if you eat this genetically modified banana, somebody will have heart illness or kidney illness. So there is no direct correlation between GMOs. And what also should also think that plants are there in open environment. They're already getting a recombinant, the birds can take pollen from one plant to the other plant. Broccoli came out from natural recombination, natural evolution. Broccoli was never there before. So some kind of mutation, some kind of recombination happened, and it gave rise to broccoli, which is very rich in various kinds of nutrients. So so those are so again, I don't want to say that biotechnology is has positive impact or a negative impact, but you can think about it like in terms of vaccines, medicines. Now people who have cancers, they can live more, but depends when they're diagnosed. So there are positive impacts, there could be controversial impacts, that's up to you. But as a student, as a teacher, we should be informed. And question number 6, discuss the main issues of biotechnology. So when the technology is increasing, people are having ideas, it has to be regulated, because we don't want to do something unethical. We don't want to copy somebody's hard work and make our own. So it has to be regulated. So we'll study that in the end. SLO number 7, demonstrate the understanding and ability to identify, evaluate current issues of biotechnology in making informed personal and societal choices as citizens. So same thing, to keep ourselves as aware. And we will discuss various kinds of articles as we move forward. And then critical thinking. On one hand, you look at the positive impact, then also you should look into negative side or try to debate an issue. And ninth is to insert a writing intensive assignment of this course. So before I go into, I'll just try to discuss or show you what book is needed. So the book that is prescribed in this syllabus and this course is, let me share with you, is Biotechnology Demystified. This is the book. This is available on Kindle at this price and paperback. So I teach most of the things from this book. Sometimes to give a better presentation or better figure, I try to borrow images from Google, but mostly I follow this book for all my slides. Most of the slides, 90% of the slides are from this book. So let's go back to the syllabus. So I would directly go to the structure of this course. There will be assignments every week. Every week we will cover a chapter. Assignments will be your handwritten assignments. And there will be, this assignment will be for 20%. The assignment will also include discussion board, which will be 10%. Quizzes, possibly weekly or bi-weekly. Total quizzes will contribute to 10%. Midterm, 20%. On the day of final exam, what is expected is a term paper. I will post the guidelines for the term paper by next week. And I'll also put a YouTube video explaining the term paper and what is expected, and the final exam, 25%. So this is a total how your this course is structured with the point of, from the point of grading criteria. Now let's look at the topics. So the first chapter will be to review the syllabus, the first day, but we are just discussing the syllabus today. From week 2, we'll start looking into various chapters, which will be on macromolecules of life, so and so forth. This will be followed by third week, introduction to living things, cellular basis of life, that will deal with the structures of a bacteria and a human cell and a plant cells. Then how does cell talk to each other? What is the role of lipids? Then we talk about cell cycle and how does cell divide. And there are two ways by which a cell can divide. Uh, a normal cell generally divides by mitosis, 1 to 2, 2 to 4. But there are special cells that are responsible for reproductive functions. They undergo meiosis. So we'll look at mitosis, meiosis, we'll look at the differences between them, and so forth. Um, Week number 4 is the DNA, the blueprint. We talk about the function of DNA, like replication and then transcription and translation. In week 5th, we talk about Mendel. In Mendel, we talk very briefly about his important theories, especially principle of dominance and codominance, or recessiveness. We talk about this in week 5. In week 6, we move into applied aspect. We talk about, we learn about recombinant DNA technology. In the week 7th, we talk about, or we learn about protein biotechnology. Learning about protein is very important because all these vaccines that we are having, what do we get? We are getting mRNA from these companies into our body, and these mRNA, they make protein, spike protein of COVID, and that generates our antibody response. And why did we generate this? Because this antibody response serves as a memory to encounter or to defeat the virus when we encounter them in real life. Then week 8 is about biotechnology in agriculture and then animal biotechnology. Let's come down to week 12. We talk about stem cell technology, types of stem cells, cloning of stem cells. Stem cells have become very important, especially with the increase of diseases where it causes organ failure. It could be kidney failure. It could be a lung transplant because of COVID. It could be a heart transplant. So stem cells are very important because if we know how to use stem cells for therapeutic purpose, we can overcome the need, the scarcity of organ donation, because it's extremely painful to wait for new organs from somebody who is unknown. There's a long line. So stem cells can do wonder in treating organ-based diseases. Then we talk about biotechnology in medicine, disease diagnosis, gene therapy, all these small topics in week 13. In week 14, we talk about biotechnology regulations, patent and biotechnology, so and so forth. So we could have a quiz every week for every chapter, and so and so forth. So now I stop my recording, and I look forward to your questions.